Yeah, we're learning more about Sam than we want to know.
Say what?
We're learning more about Sam than we want to know.
That's right.
He's demented and horrible.
Yeah. When I was talking about killing Jamie, little did we know that Sam was such a psychopath.
Sam is actually worth it.
Ow!
All right. We are surprisingly enough right on schedule here at the beginning of the second to last week of school. So
That was so funny.
I don't know what to say about that other than that.
Is that another tester?
What's that?
When, right?
Just the final. Yes.
When is our final scheduled for?
Oh, I have no idea. Sometime during
That's
finals
right.
week, and it's on the finals schedule.
I think it's
Nice.
Wednesday.
Oh,
That's,
I think it's on Wednesday at 11
that's
o'clock.
really good.
That
Maybe.
sounds familiar.
Maybe one.
So that's hopefully
Amanda, I will show you
Thank you.
how you can find this information yourself. You guys know about the intranet, Joule Central?
No, Jewel Central did not teach me.
Joule Central, central.joule.edu. Under students, there is a section where it says exam schedules. Click on exam schedules. Fall exam schedules, or what, the exam schedules for 2021 and 2022. So you just go through here and you find your section. We are block E, 12:30 Monday, Wednesday, Friday, so we are Wednesday at 11, so somebody who said that was absolutely correct.
Oh yeah, that was her.
I think it's also listed in the syllabus, but I generally go and look it up to put it in the syllabus, and then I forget about it until the week before
Yeah, I
finals.
looked at the syllabus, I didn't find it, I could have
No.
completely, like,
Yeah,
missed
it,
it, but,
it might be, it might not be. There's a central repository for these things.
thank you.
While you're on Joule Central also, if you need to know, for example, for planning next year, when spring break is, you can go to the important dates. It tells you when advising days are. It tells you when you can register depending on your status. It tells you when spring break is. All the important dates during the semester are also on that website. So
Fun fact of the day.
you can plan ahead. I use these two things frequently when I'm putting together a syllabus to make sure that I'm scheduling dates for classes correctly and stuff like that.
Thank you.
Sure, no problem.
What is
Part
it?
of college
What
is just figuring out systems and how systems work, and systems can be opaque. In the old days, we used to submit our course proposals for our course schedule for the next semester on these green forms. They were green forms, and you wrote by hand what your courses were and when you wanted to offer them. And then we went to an online system, and for years after the online system came online, the registrar would send us a request to fill out our course proposal sheets. And they were like, please fill out your green sheets. And finally, one day I got tired of it. I'm like, they're no longer sheets, they're no longer green. If you weren't here back when they were sheets in green, you have no idea what you're asking for. So can we just call them course proposal forms and be done with all the arcane knowledge that you have to have historical history about, historical knowledge about it to understand what we're supposed to be doing with that. So yeah, systems are sometimes hard to figure out, and they're especially hard in a place like Joule that's been around forever and has difficulty thinking about the other, that new person.
Yeah.
So, yes, ma'am.
Are we having lab finals?
No lab. Nope. You will have, listen up, you will have assignments related to lab that are due during that week, but we will not meet for lab. We will have lab the last week of classes, which was when you guys will do your oral presentations. Oh, speaking of lab, tomorrow when you come to lab, bring two printed copies of your lab report. Sorry.
No, that's cool.
Ooh to 2 copies or ooh to the lab report?
You to the lab report.
Okay.
Is that just your lab or our lab too?
Both labs.
He's
2
never
copies.
even mentioned ours.
No.
Well, he's probably going to mention it at the beginning of class today.
Where he at?
I don't know. It's not my day to keep track of him, and he repeatedly chews his tracker out of his arm, and we just can't get him to keep it in.
My God.
On his head.
Oh,
We're gonna cut him open and put it in his torso so it's harder to get rid of.
Jesus.
All right.
I like the way you think.
The last case study that we're working on relies on quantitative genetics. We've already covered quantitative genetics, so part of today is going to be review, and then we're going to go beyond what we learned before about quantitative genetics. So the first part is going to be review, the second part is going to be expansion. So you should be able to describe what quantitative traits are. You should already be able to do that because you had to do that on test 2 of this semester. You should be able to calculate the evolutionary response to selection for a single trait. Once again, you should be able to do that because you've already been asked to do that on a previous test, exam 2. You should be able to illustrate what positive and negative genetic correlations are. That's new material. We haven't talked about genetic correlations yet this semester, but we will today. You should also then be able to calculate the evolutionary response to selection for genetically correlated traits, so dealing with 2 or more traits. It's the same idea as before, it's just another little added layer of complexity onto it. We'll use a little bit of matrix algebra, but we'll show you how that works. And then you should be able to explain how genetic correlations affect how evolution can proceed. Because sometimes evolution changes a species in a way that you don't expect, and it's because the trait that is under selection is actually correlated with another trait that is not necessarily under selection, or is under selection in a different way. So remember that we use quantitative traits, we use quantitative genetic to study these quantitative traits. Who can give me some examples of what a quantitative genetic trait is? Anybody? Well, athletes and half-mates. Athletes, half-mates. Examples. Back toppers.
So quantitative has to do with numbers, right?
Yes.
So measurables, right?
Yes.
Something like that. So
There are all sorts of things that can be measurable.
for sure, yep. So my group, what do we think?
Would height be quantitative? Height?
Yes, height would be it.
There you go. Yeah.
What's up, Garrett?
Another example of quantitative trait. Hair color, actually it is quantitative because it's determined by multiple genes, and you could actually, in theory, develop the scoring method for doing color. In the same way that eye color is also a quantitative trait. We don't normally think of it in terms of numbers, but it's kind of the intensity of color. Wheat grains vary from pale yellow to white all the way to dark red. That's another quantitative trait. So a lot of times we think that's why I hedge when Sam says numbers, is because we do generally think of them as numbers, but we can also think of them as other non-numerical things that we can put on some sort of continuous scale. And hair color and wheat, seed color and eye color would all be these things. Seed color, when it's not a binary thing like in peas where they're either yellow or green, that's not a continuous scale of variation, that's a discrete scale of variation. Seed color, eye color, what else did we say? Oh, hair color. Now, what's the problem with hair color? Okay, so people can dye it, that's an environmental effect, but what else happens with hair color? Yeah, it changes with age, and so it's one of these sets of genes that is affected by the developmental stage that the individual possesses that particular trait. So I got something for pre-med. Did I get anything for athletes and athletes?
Nope.
Oh yeah, the gray hair.
There you go. Genius. Why would I give it?
Okay. So these types of traits are undergirded by multiple genes. Each of those genes contributes something to whatever the trait is. In the case of height, the more dominant alleles you have, the taller you are. Just keep that back there for now.
Yeah, more points coming our way.
So the math, remember that the math that we use to look at quantitative genetic traits is ultimately rooted in Mendelian characteristics, in Mendelian genetics. Things like flower color and pea color and pod shape and stuff like that in peas is the same kind of genetics going on in quantitative traits. It's just that there are more of them contributing to a particular trait. And so a lot of times the goal of quantitative genetics is to show how selection acting on one generation will produce genetic change in the next generation. So you'll recall stuff that I've already said. Oh, I've already said all this. Here's some examples of other things. Ear length in corn, crop yield, fat content in meat, height, sprint speed. Just so many things fall under this category. One of the reasons we study quantitative genetics is oftentimes the kinds of traits that are quantitative genetic traits are traits that are of interest to biologists. So if you are a corn farmer, you might be interested in how many kernels of corn are on your corn cobs, because the more kernels of corn on your cobs, the more kernels that you get per plant. If you have X number of plants per field, then more kernels per cob means more bushels of corn to take to market. And so these are all things that have interest to evolutionary biologists, mainly because they have interest to other people as well. So we went through this exercise earlier when we looked at 3 traits, 3 genes all contributing to a particular trait. Each dominant allele contributes one thing, one unit to that trait. And we did the cross. Do you recall this? Is this ringing any bells? The cross ended up in this big, huge mess of genotypes. But we could just reduce that to the number of dominant alleles. And when we do that and we count up how many individuals have 6 alleles versus 5 versus 4, we end up getting this distribution where very few individuals have either no dominant alleles or 6 dominant alleles, and it's this normal shaped curve that comes out of that simple genetic cross. So by virtue of the normal distribution, we can then do genetics by using mathematics rather than by paying attention to the genes themselves, which is also nice because you don't even need to know what the genes are or where the genes are located in the genome to work on them. All you need to do is know what the shape of the distribution of the trait is. So when we do this, we can calculate heritability. Who can tell me how we calculate heritability? Slope of a line of a regression between what and what?
Parent and offspring.
Parent phenotype and offspring phenotype. That's one for the mitochondriacs, and one for the sport specimens. So that's where we get heritability. It's the slope from a parent-offspring regression. Offspring phenotype regressed onto parental phenotype. The strength of selection we generally have to measure in the field. The strength of selection is the difference between the parental population pre-selection and post-selection. So we look at the parents before selection occurred and the parents after selection occurred. We look at that difference in the population mean for that trait, whatever that is. And if we know what the shift in the trait is in the parents, that shift in trait gets transmitted to the offspring through heritability, and that's what leads to the evolutionary response to selection. So we can do this with any trait that we can basically put some sort of quantitative number on. Remember that little Butch Brody was working on Thamnophis aerodynamic, one of the members of the genus Thamnophis that Thamnophis tertalis is also in, but he was interested in them from the standpoint of how stripy they are. So in this population in Oregon, the snake species Thamnophis aerodynamic is what we call polymorphic. Some of the species are really stripy, and some of the species are really checkered-y. And there's a continuum between extremely striped and extremely checkered. And he invented a scale by which he could count up characteristics along the body of the snake, and he derived a formula for basically putting a number on how stripy a snake is versus how checkered a snake is. He was also interested in sprint speed, so he would race snakes down racetracks and measure their speed. And when he was doing these things, he also noticed that some snakes would tend to change direction while he was chasing them down the racetrack. And when he did this, he could count the number of times that the snake would reverse itself while being prodded down this racetrack. And so he had these snakes. The snakes give live birth. So if you catch a pregnant female, you have not only the parent, and you also have all the offspring. So he could measure stripedness in the parent, he could measure stripedness in the offspring, and he could look at the correlation between these two things. And once again, the slope is the heritability. So in this case, the heritability would be 0.707. And if this was the average size, or the average stripedness rather, of the female snakes, you could predict what the average offspring stripedness would be based on this regression. But if something happened that made snakes stripier over the course of a generation, you could also then predict what the effect of stripedness in the offspring would be, once again by relying on this correlation. And this delta Z is also what R is. Delta Z is just the change in the trait value, which is also the evolutionary response to selection. You will see both of these in the literature, so it's good to know that both of these mean the same thing. And so we could easily look at selection. So let's say that the mean stripedness value changed by 2.29 units, and the heritability is 0.707, then we would expect the stripedness of the offspring to increase by 1.62 stripe units, whatever that means according to his calculation. And so calculating the evolutionary response to selection is pretty easy if you know heritability and you know the strength of selection. There are other ways of kind of checking your math on this. This is the equation for the line, 0.707. This would be multiplying 0.707 times the new value of the offspring, or of the parents rather. So if we took 2.29 and added it to the pre-selection, this is the pre-selection average stripedness of the parents, the post-selection average stripedness of the parents. So this is where the 2.29 selection strength comes from. If we took this post-selection value, multiplied it times the slope, added it to the y-intercept, from this equation here, it will predict for us where that average offspring stripedness will be. And so when we do that, we find that the average stripedness of the offspring would be 12.57. The average stripedness before selection would have been 10.95. 10.95 taken from 12.57 is 1.62, which is the response to selection. So there's a variety of ways in which you could approach this. The simplest way is just know what heritability is, know what the strength of selection is, and do the math that way. I'm not ever going to ask you to do the rest of this stuff. This is just included so that you can see, hopefully, the relationship between heritability, the strength of selection, and the response to selection. And it's always the heritability. It's not R square. Don't confuse R square in this. What brings the offspring population up is selection multiplied times the slope of this line. That's what raises the offspring trait. If heritability was 0, this would be a horizontal line, and no amount of selection would result in a change in offspring phenotype. The steeper this line is, the more evolutionary change you can expect. The more shallow the line is, the less evolutionary change you can expect. So the part of this slide to pay attention to is this upper part. The other stuff is just internal validation that what I'm telling you about this relationship is actually true. If you can do that for stripes, you can do that for reversals. The slope is different, so the heritability is lower. Was 0.707 for stripedness. It is 0.245 for reversals. Does this mean that evolution is going to go more slowly or quicker? You might think more slowly because the heritability is lower, which means the slope of the line is lower. But what's the one thing that can make it go faster? The strength of selection. If selection is really strong, it'll go faster even though it has a lower heritability. but we have moderate levels of selection, and in this case, selection is selecting for fewer reversals rather than more reversals. And so we would see the change in the offspring to be a negative change. We can do the math. You can see reversals is down around 2, goes down to what, about 1.3, something like that. So it was 1.35, I'm sorry, it was 1.94, it went down to 1.35. That's a selection differential that is a negative. That multiplied times the evolutionary response to selection gives you a negative, that multiplied times heritability gives you a negative evolutionary response to selection. And once again, this is just showing how you can come to the same answer via a different route. So selection can increase a trait value, selection can decrease a trait value. It all depends on what direction selection is pushing the population. So we're mainly talking about directional selection, but remember that we can also have stabilizing and disruptive selection as well. This tends to move a population one direction or another direction phenotypically. This basically keeps you at the same population mean generation after generation, but variance is decreasing. This tends to keep you at the same population mean from generation to generation, but the variance is increasing, getting wider apart, because more of your population is distributed out in these two modes, away from the mean, making variance higher. So there are the labels. This, once again, this is all review so far at this point. So we have stripedness and we have reversals. We can think of these two things as being disconnected from one another, not related to one another at all, or we could think of these things as being correlated with one another, such that something that happens to one trait forces something to happen in the other trait. Under what conditions might you expect two traits to be linked to one another in this way, such that as one thing happens to one trait, it also happens to the other trait? You guys remember from high school a term called linkage? You guys studied genetics in high school? Part of the biology class? Bruce?
Didn't go over that one.
Didn't go over that. So sometimes on a given chromosome, a gene for trait A will be on one section of the chromosome, and then you have to have another chromosome type, and trait B will be on another chromosome. And in this case, what happens to one trait isn't tied to what happens on another trait, because those traits happen to be on different chromosomes. But sometimes, all throughout the genome, there are traits that lie right next to one another. And what happens to trait A will also happen to trait B, because they happen to be linked to one another on the same chromosome, as opposed to being inherited separately on different chromosomes. And this is one way in which traits can become correlated with one another. The further away you get from trait A on this chromosome, the more trait B will start to behave as if it's on its own chromosome, because these things tend to break apart during synapses, during prophase 1 of meiosis, when gametes are formed, such that genes that are far apart on chromosomes tend to behave as if they are on different chromosomes, whereas genes that are located close to one another on a chromosome tend to get inherited together. So that's a common way in which genes can be linked to one another. What's another way that genes can be linked to one another? Or traits can be linked to one another, actually. You guys know the term pleiotropy? Anybody? Look up what pleiotropy is. And stop playing jigsaw puzzles.
gene or two or more apparently unrelated effects?
Okay, so in some cases you have a single gene that basically controls more than one trait. So this is another way in which two traits can be correlated. It's because they're under the same control of a single gene. In you guys, you have growth hormone. There's a gene for growth hormone. Growth hormone is a protein. And the amount of growth hormone that you have affects a lot of aspects of your phenotype. One of the main effects is that it controls how long your long bones stay cartilaginous so that you can increase long bone growth. So if you're really tall, you either produce more growth hormone or you produce growth hormone for a longer period of time. If you're short, you're producing less growth hormone or you're producing it for a shorter period of time during development. There are some people who have a genetic disorder where growth hormone production doesn't shut off after puberty and they just continue to grow their entire lives. And their long bones get longer and longer and longer, while other bones in their body stop growing because they're not governed by the same developmental pathway. At the same time that growth hormone is affecting your long bones, though, it also affects things like your larynx development, so how big your larynx gets. And so some genes have multiple effects on multiple traits is another way that two traits can get connected to one another other than just being on the same chromosome. So these physiological connections oftentimes lead to connections. In this case, we're looking at a morphological character and a behavioral character. And so those things don't probably have a physiological underpinning, they have some other underpinning. They're either on the same chromosome, or it's possible that selection could lead to these two traits actually being selected for in coordinated evolution. So this is Big Butch. This is at the University of Texas at Arlington. This is him running a snake down a racetrack. This is a more checkered snake going down a racetrack. It's lined with AstroTurf and it has infrared sensors, and as the snake breaks the infrared sensors, computer registers the speed. I have a similar racetrack, only smaller for geckos, and I use lasers in mine rather than infrared, because I also race my geckos at very high temperatures, and infrared tends to not be very effective under those conditions. So, if a trait is genetically coupled with another trait, when selection acts on one trait, it may affect the other trait that it is coupled with. So this is what we mean when we talk about a genetic correlation. So how do we determine if traits are genetically correlated? Well, the first thing we can do is we can treat it kind of like a heritability. We can look at the trait in the parent, trait 1 in the parent, and trait 2 in the offspring, and see if these things are correlated. If they are correlated, it means that the genes that are affecting reversals are the same genes that are affecting stripedness. And if that's the case, then you have a genetic correlation. And so the possibilities are that there could be a positive genetic correlation such that the more reversals you have, the stripier you are. You could also have a negative genetic correlation where the more reversals you perform, the less stripy you are. Fewer reversals means more stripe, more reversals means less stripe. And then of course you could have a slope of 0, in which case there is no genetic correlation at all. Well, we can take Fisher's breeder's equation that basically looks at selection multiplied times heritability to project what evolution is going to do, and we can expand it to include more than one gene. So how do we do this? More than one trait, rather. Slope a little bit there. So in this case, we want to know what is the evolutionary response to selection of trait 1, and we want to know what the evolutionary response to selection is of trait 2. So we're going to just put those guys in a vector. And the evolution of a trait can be influenced by selection. And so our symbol for selection on trait 1 is going to be beta 1. Selection on trait 2 is beta 2. So these are the equivalent of S in the single variable. This is the equivalent of R in the single variable. And then we have this big thing G, which is the equivalent of heritability. But we have now two kinds of heritability. We have heritability for trait 1, which we're just going to say it's the correlation between trait 1 in parents and trait 1 in offspring. That's heritability for trait 1. We can also have the heritability of trait 2, which is simply the correlation of trait 2 in parents to trait 2 in offspring. But then we also have the situation where we can have trait 1 in parents being correlated with trait 2 in offspring. That would be G12, but that's the same as saying trait 2 in the parents and trait 1 in offspring. So these two things are the genetic correlations. Or covariance, they're also referred to in the literature as covariances, because they covary. Trait 1 covaries with trait 2. They will be exactly the same magnitude, but we get at them by regressing trait 1 on trait 2 or trait 2 on trait 1 in the parents and the offspring. Yeah.
So is the first number the parents? The second number is the offspring?
Actually, we're thinking about it in terms of trait 1 and trait 2.
Oh, okay.
So this is the heritability of trait 1, this is the heritability of trait 2, this is the genetic correlation between trait 1 and trait 2. These subscripts refer to kind of how the math works, which I'll go through in a minute. But these are the same. So you can measure trait 1 in parents and trait 2 in offspring, or you can measure traits 2 in offspring or trait 1 in, trait 2 in parents and trait 1 in offspring. And in reality, the way you do it for really is you run both of these regressions and you take the average of it. It's called geometric mean regression, and that's how you actually arrive at what this number is. But this number is always the same as this number. And once again, those correlations can be either positive, they can be negative, or they can be flat. Flat meaning there's no genetic correlation, and this would be 0. This would be positive values here and here, and this would be negative values here and here. They would be the same magnitude. Now, to get what the evolutionary response to selection is, you just use this equation. This is actually an equation. This is a multiplication right here. So to get the evolutionary response of trait 1, you take G11 and you multiply it times beta 1, and then you add that to whatever G12 is multiplied times beta 2. So what this basically says is trait 1 evolves. And it evolves in response to direct selection acting on trait 1, but it also evolves via correlated selection of trait 2 acting on the genetic correlation between trait 2 and trait 1. Both of these things contribute. In the same way, trait 2 can respond to selection due to direct selection acting directly on trait 2, selection on trait 2 acting on the heritability of trait 2, but then you also have to consider what's happening to trait 1, where selection may be acting on trait 1, and that affects trait 2 as a result of this genetic correlation between the two of them. Okay? So that's just how the math works. You take this times that plus this times that. Then you take this times that plus this times that. Matrix algebra, it's not complicated. Yeah. Okay. Any questions about that? This is what it looks like. You can think of it as row and column. So the first number is the row, the second number is column. G11, first row, first column, second column. Second row, first column, second row, second column. That's just how the numbering in the G matrix works. If we do this for reversals and stripe, reversals, this is heritability of reversals. This is the genetic correlation between reversals and stripe. This is the genetic correlation between reversals and stripe, and this is the heritability of stripe. we can actually measure. Oh, we've already done this. We've got the genetic variance or heritability of reversals. We've got the heritability of stripedness. But what we need now is we need the genetic correlation between reversals and stripedness. And in this case, it's a negative genetic correlation. The more reversals you perform when you're being chased around in a racetrack by a big bearded man, the more you do that, the less stripy you are. And then we can look at selection on reversals in this case and stripedness in this case. And once again, selection acts on trait 1 directly. So if selection is pushing reversals to be higher in the parents, it will also be higher in the offspring because this is the heritability of reversals. If selection is acting on stripedness, it will also increase in offspring because there's this positive heritability. But, now this is a color-coded version. Selection, things that apply to trait 1, things that apply to trait 2, genetically. This is just a repeat of what I've already told you up there. So if we do this with stripe and reversal, we can actually do the same thing, just in stripe and reversal terms. And then what I want to get to is I want to get to actually thinking about this in terms of the evolution of reversal and the evolution of stripe. So let's say that we start off with a population where you have a lot of reversals, but it's a pretty checkered population. So let me do this. This might be easier. If we reflect this dot down here, it reverses a lot, and if we reflect it over here, these are relatively checkered snakes. Okay? Now, we can ask ourselves what happens to this population from one generation to another generation by looking at all these different contributions to selection and the underlying genetics. So, if we have selection acting on reversals, but the selection is not particularly strong, selection on stripe is considerably stronger. We might expect stripedness to evolve very quickly because selection is acting on it more strongly than the selection that is acting on reversals. But of course, what can influence the speed of the response to selection? Is it just selection that matters? Or do we also have to think about something else?
Heritability.
We also have to think about heritability. So we need to not only know what the heritability is of, in this case, stripe, we also have to know the heritability of reversals. So here we have selection that is relatively weak acting on relatively low heritability. Is that thing going to be responding quickly or not? It's going to be responding slowly. Here we have strong selection acting on something with a relatively high heritability. Would we expect quick evolutionary change or slow evolutionary change? We would expect quick evolutionary change. Large heritability, large amount of selection. Are these the only two things that we need to worry about, though? No, because we also have this high selection can influence the evolution of this trait through this negative genetic correlation. Is that negative genetic correlation on the high side or on the low side? High. They range in the same way that heritabilities do. They range from 0 to 1. So this is on the high side. So we might see this is positive selection acting on this heritability. So we might expect trait 1 to be responding positively to that selection in the same way. If we just look at selection and heritability, we have strong positive selection acting on heritability. So we might expect a positive movement of the evolutionary response to selection. But this high selection acting against this negative genetic correlation may drag this to the point that you actually see reversals decreasing from one generation to the next. in spite of the fact that there's positive selection for reversals. But it all depends on how the math works out. So let's just do the math. We're going to take 2.45 times the selection, this times the selection on this trait, this times that selection, and this times that selection. And when we do the math for all of that, what we find is that this is this, this is this, this is this, that is that. And when we add those across to get the evolutionary response to selection, we actually find that reversals actually decrease while stripedness increases. Stripedness increases because it already had strong selection acting against high heritability, but then the genetic correlation is multiplied times a relatively weak selection on the other trait. Reversals, so stripedness tends to still increase. And so in this case, evolution is going just the opposite direction that you would expect based on the selection acting on that trait. So this is one of the first important findings to think about, is that genetic correlations can reverse the direction that selection is actually acting on a trait because of selection acting on another trait that is genetically correlated to. We can think of it graphically by going back to this figure here, and we can say to ourselves, what happens to this population as a result, first, of selection acting on reversals? So if we look at selection acting on the heritability for reversals, it goes up a little bit. If we look at selection acting on stripedness, stripedness moves a lot further because the heritability for stripedness is high and selection is high. But this doesn't include anything about the effect of genetic correlations. So we can look at what the effect, so this is where they would be if we didn't have genetic correlations. So we can look at the effect of selection on stripe on the directional movement of reversals, and it's dragging it back. And then we can look at the effect of selection on reversals and how it affects stripe, and it drags it down. But the degree to which selection on stripe drags reversals back is much bigger than the effect of selection on reversals dragging stripe back. So that in the end, we end up over here, when if there was no genetic correlation at all, we would have ended up over here. Both reversals and stripedness would have increased, but in this case, reversals decreased some, and stripedness didn't increase as much as it would have if it hadn't been for that negative genetic correlation. So R is this direction for these two. And I think I have it. Oh, I do have it in R in this figure. This would be R1 or R2. R for stripe and R for reversals. So this is just another way of thinking about what that equation is doing in a space that is one character versus another character in terms of their value. Reversals from very few reversals to a lot of reversals, checkeredness to stripedness. Questions about this? Math is a little complicated, but it's nothing that you shouldn't be able to work through with a little practice. And we're actually going to give you a homework assignment where you'll practice this. Questions? Okay. What do we have here in terms of selection on reversals and stripe? Same as before, right? Nothing's changed. So in this next scenario, what has changed? Are the heritabilities the same? 0.245 and 0.707?
Yes.
They are. So what has changed?
The, before the heritability.
It's genetic correlation. They're still negative, but are they as negative as they were before?
No.
No, they're weaker. So we can do the same thing. We can look at the effect on reversals of direct selection acting on reversals. We can look at the correlated effect of selection acting on reversals through selection on stripe. We can look at the direct effect of selection acting on stripe, and we can look at the correlated effect of selection acting on reversals that then affects stripe. And what we see in this case is because that negative correlation is less strong, the increase in stripe is actually more than it was before, and the decrease in reversals is less than it was before. It's closer to 0 now, rather than being of a higher magnitude than it was before. This is the equivalent of, these things are the same. This is the effect of directional selection. But in this case, the effect of correlational selection is lower because the correlation between the two traits is lower. So there's less of an effect of dragging back reversals, and there's less of an effect of dragging down stripedness. Reversals still end up being slightly lower than they were before, but it's just by a little bit rather than a big amount, because of those changes in the magnitude of the negative genetic correlation. What's happened in this case? Yeah, the selection on stripedness is now much stronger. Here's the genetic variance covariance matrix. This is the same scenario that we had before. What do you expect the outcome of selection to be under this condition? Selection is now stronger on stripedness, but it's the same as it was before on reversals. The magnitude of the negative genetic correlation is the same as it was in the first scenario. What do you expect to happen to stripe and reversals under these conditions? Without doing the math, just intuitively, what do you expect?
R will be higher.
What's that?
R will be higher.
For which one? Yeah, so R will be higher for stripedness. What's going to happen to the evolutionary response of reversals? It's going to be of greater magnitude, but that greater magnitude is going to be negative, yeah. So do the math. And what we see is this huge change in the evolution of reversals going the opposite direction of what direct selection on reversals would indicate. And it's because you have a lot of selection acting on reversals through this genetic correlation. It's this scenario. This is the same as it was in previous graphs, but this is much higher. As a result of selection on stripe being strong, this is a much longer negative arrow, dragging reversals way back. This is the same amount of selection acting on the genetic correlation for stripe, so it drags it back the same amount. So stripe increases more because it's being dragged back the same as it was before in the earlier scenario. Here's the same selection that we had before. What's different about this genetic correlation? It's positive. What do you expect to happen to stripe and reversal in this case?
They won't get dragged back as much.
Neither of them will get dragged back. They won't get dragged back at all.
They dragged forward.
They'll get dragged forward because selection acting on trait 1, reversal, selection acting on reversals directly moves it forward, and selection acting on stripe also moves reversals forward. Selection acting on reversals moves stripe forward, and selection acting on stripe moves stripe forward. So these actually make evolution move faster than you would expect if the genetic correlation weren't there. So this is the direction now. .068, it's now in positive territory. .077, it's in positive territory. The directional effects are the same as they were before. I've drawn them opposite one another because of the next thing, but they're the same, the arrows are the same lengths as in the first scenario. But what happens with correlated selection is correlated selection just drives stripedness more stripy, and it drives reversals to more reversals. And so you end up at a much higher level of reversals and at a much higher level of stripedness than you had if there was not this genetic correlation present. So what's the effect of genetic correlations? They can, what happens in this case? Is the evolutionary response proceeding faster in the positive direction, faster in the negative direction, or there's no effect? Faster in the positive direction. So genetic correlations can speed up evolutionary change in the positive direction. What else can they do? Yeah, they can reverse the effect of selection. So selection can be acting positively on a trait, and it can be moving in a negative direction. Or evolution can be acting negatively on a trait, and it will move in the positive direction. So it can reverse the direction of evolutionary change from what selection would indicate. And then the other thing that it can do, if it's just right, is it can just cancel out the effect of selection altogether. Imagine if this is a contrived case. Imagine if heritability of trait 1 was 0.25, the heritability of trait 2 was 0.25, the genetic correlation between the two of these things was negative 0.5, and selection was acting positively on trait 1 at this level and positively on trait 2 at this level. These two things would basically end up as 0. No evolutionary change at all, because the strength of selection acting on this trait is the same as the strength of selection. Is that right? No, sorry, I have to do my math right. Strength of selection driving this trait positively is the same as the strength of selection driving this trait negatively. Same magnitude, they just cancel one another out. So you don't end up with any evolutionary change on either of these traits, in spite of the fact that you have selection acting to move the average values of these two traits forward. And it doesn't matter, it doesn't have to be low, it can be very strong selection acting on these traits. And still you're going to get them canceling one another out because of the negative genetic correlation. So, and we just kind of answered these questions. So negative correlations can cancel out or slow down evolution. Positive correlations can speed up evolution. And so then the next question is, how do these correlations come into being? So we've already talked about a couple of ways in which these correlations occur. One is that they occur on the same chromosome, so what happens to one gene happens to the other gene. It could also be that the thing that affects one trait is also affecting the other trait because they're controlled by the same gene. But the other thing that can happen is that selection can tie them together. So in this case, what Brody did was he caught a bunch of snakes, he scored them for stripedness and he scored them for reversals, and then he let the babies loose out on his grandmother's land. He did this at his grandmother's house in Oregon. And the nice thing about these garter snakes is that they exist in hibernacula, and when the parents come out in the springtime, the females are already gravid, they're already carrying offspring. So he could go there in the spring, he could collect all the females, he could measure the heritabilities of the traits because he had females and offspring, then he could let those offspring loose, but then in the fall, all those snakes come back to the same place where the parents came from, because they all den up in things called hibernacula, and they overwinter in these underground spaces all together. They breed over the winter, they emerge pregnant the next spring, and they go through the whole process again. So he could let loose the babies, and the babies would come back to the hibernaculum in the following fall. And so he could look at who survived and who didn't, and relate that to their reversal score and their stripedness score. And what he found was that the snakes that had the highest survival were snakes that were striped and did not reverse themselves. The next highest survival were checkered snakes that reversed themselves a lot. Snakes that did less well were checkered snakes with low reversals, did not reverse themselves very often. And the worst performers were striped snakes that reversed themselves a lot. If you have a race car, what do you paint on it?
Stripes?
Yeah, you paint it with spots or checks?
No.
No, why?
Stripes are aerodynamic.
Stripes are not aerodynamic.
They look cool.
Stripes are cool.
Stripes take advantage of a sensory bias that humans have. And it's a sensory bias that most vertebrates have. Stripedness on a body that is moving makes that thing look like it's moving faster than it actually is.
Aerodynamic.
It's not aerodynamic. It's only
No,
an
that makes
optical.
sense. That makes sense.
It's only an optical illusion. Now, if you want to get stopped for speeding more often, put stripes on your car, because it will make you look like you're going faster than you actually are. If you're a snake and you're striped, as you're moving, it makes you look like you're moving faster than you are. So if a predator is trying to attack you, it's going to oftentimes miss. And the reason is because your eye elongates that stripe. It moves it out in front of the snake, and it also trails that stripe behind the snake in the way your mind perceives that color pattern. What happens when a checkered snake, don't do that to me, sick. He's waving his stripes are out. His stripes not.
Moving so fast.
What happens when a checkered snake is moving? What happens to all that checker when it's slithering through the grass?
Oh, it's more like camouflage.
It's not camouflage when it's moving.
It looks weird. It's confusing.
If a checkered snake is speeding through the grass, what happens to all that checkeredness?
Comes like a wave.
It just blurs together and it looks like a uniformly colored snake. What happens as soon as that snake stops?
It's obvious.
Will it be obvious?
It will be non-obvious.
It will be non-obvious because the checkered breaks up the pattern of the body. It becomes camouflage. You finally get to say camouflage.
Yeah.
You finally get to be right. Excellent. So if a checkered snake is moving quickly through the grass and stops and turns the other direction, what's happening with the visual image of the predator? Got a solid looking snake going through the grass. It looks like a line in the grass. It's extending that line out in front of the snake and extending that line back behind the snake. If it stops and turns around though, and then sits there,
Well, then it'll see where it's at. It won't look as fast.
well, it's not looking fast at all. Your eye was following it out. It turns around and stops. It's checkered now.
You're looking for one color.
Oh.
You're looking for one color,
I would've gone straight.
and your eye has gone past where the snake actually stopped because your eye was following it, and you had to come back to it. By the time you get back to it, it has stopped and blended in and become camouflaged. So checkered snakes that reverse themselves tend to survive pretty well. Striped snake, speeding away from you, stops, turns around, your eye follows it back, but follows it out, but then it comes back because you don't see the snake anymore, it comes back. What do you see?
The snake.
You see a striped snake. Because the stripedness really stands out because it's lines, and nature abhors lines. Look around, straight lines are man-made. Curved lines are sometimes man-made, sometimes natural, but what they do is they don't obscure the outline of the snake. They call attention to the outline of the snake. Striped snakes that reverse, sorry, I said checkered snakes that reverse a lot do well. Striped snakes that reverse a lot, guess what?
Poorly.
Dead.
They get killed a lot. Striped snakes that just haul ass through the grass, hard for you if you've ever tried to catch a snake with a stripe moving through the grass. A lot of times you grab for its tail and you miss because you're grabbing where the tail was a second ago because your eye is continuing those lines back behind the snake and also projecting those lines in front of the snake.
So you grab farther up.
Well, yeah, you learn to grab towards the head because you're a smart human. High striped snakes that don't turn around do really, really well. Checkered snakes that just keep going in a straight line,
They're still hard to see.
they're a little harder to catch, so they don't suffer much of a decline over the checkered snakes that reverse a lot, but they're still lower. But once again, that solid snake is less deceptive than a striped snake is, where you have multiple lines of striping going down the body. So selection acts to make high striped snakes that go in a straight line survive better than high striped snakes that reverse a lot. What this does, ultimately, is it results in a selection surface, an adaptive landscape that Dr. Judd will talk to you about on Wednesday, where there are two peaks of fitness and two valleys of fitness, where fitness is low, and there's this kind of saddle-shaped structure in the middle. And that's basically what this looks like. There's a peak in the far back corner, there's a peak in the front corner here, and then there are lower fitnesses in the opposite corners.
So is this disruptive selection?
This would actually be what disruptive selection looks like. Disruptive selection driving the population apart at this saddle towards a population that would have only striped snakes that go in a straight line and only checkered snakes that turn around on themselves. That is exactly what this will lead to. Wait. Oh, this is going to be Dr. Judd. Never mind. Yeah, we're at the end.
I got a question on
Yeah.
Brody. Since the paper is like a lot different, do you want like when we write it to be in the same format?
That is not the paper you're summarizing. You're doing Brody and Brody 1990.
So I just read this and oh my gosh.
You read that for the first day of the case study.
Yeah,
It was
I
in
know.
the schedule and the thing
Oh.
before Thanksgiving.
December 3rd materials?
December 3rd is when it's due. It's this Friday.
Oh, okay.
And it's there on the day that it's due, just like the other ones have been there on the day that they're due. Good thing you asked that question, Dave.
You're a striped snake that turns around a lot.
It's much shorter than that thing that you read.
Yeah, I was about to
That's
say.
an overview of the entire case study.
Okay.